Welcome back. This is day 3 of genetics, and today we're going to be talking about what is DNA composed of and how does it replicate. So we're actually going to be looking at two different things today. First, we'll look at the structure of DNA, and then we'll start talking about a process called DNA replication. Right, but before we get into that, you guys have got 2 do now questions. So go ahead and pause this video, answer questions 1 and 2, and press play when you're ready to go over it. All right, now I will warn you guys before we get started, they're still doing construction above my apartment. So if you hear like a series of bangs or thuds, I'm sorry. We're gonna get through this together. And again, I hope my ceiling doesn't collapse, but honestly, that would make for a great video. So number 1, the molecule represented below is found in living things. Which statement describes one characteristic of this molecule? Well, looking at this picture, I can see that this is a picture of DNA. And we know that DNA is known as genetic information that's found in the nucleus of our cells. So based on this information, I can figure out that the answer here is A. It is the template for the replication of genetic information. Now I will tell you guys, we're gonna see this word template again today. And when we're talking about DNA replication, when we see this word template, we're basically talking about an example or a model that the cell is going to copy when replicating DNA. So I'm going to write down it's an example or a model to copy. All right, number 2. The diagram below represents a section of a molecule that carries genetic information. There's that phrase again. We know this is DNA. The pattern of numbers represents what? Well, in this picture, I can see that 1 is pairing up with 2 and 3 is pairing up with 4. And that kind of reminds me of something we talked about yesterday called base pairing rules. Does anyone remember talking about that? We said that A always pairs with T and G always pairs with C. So based off of that information, I can figure out that the answer here is A. Those numbers represent a sequence of paired bases. And we'll talk some more about those bases today, which leads us to part A. The first thing we're going to do today is we're going to talk about the structure of DNA. So DNA consists of 2 molecules that are arranged into a ladder-like structure called a double helix. Remember, that's what we saw yesterday, where we saw DNA almost looks like a ladder that's been spread out and then twisted or kind of wrung like a towel. And so that turns it into that spiral or double helix shape. But this molecule of DNA is actually made up of millions of tiny subunits called nucleotides. And you're actually looking at a picture of a nucleotide right here in your paper. So what does each nucleotide consist of? There are 3 pieces we need to know. First, there's something called a phosphate group. And it's called a phosphate group because right here in the middle of it is this element called phosphorus. But we just need to know it as a phosphate group. All right, the second thing a nucleotide consists of is a sugar, specifically a pentose sugar, which means that it has 5 carbon molecules on it. So we can count those carbons. There's 1 right here. There's actually going to be 1 right here at each turn of this molecule. So there's 1, 2, 3, 4, 5. 5 carbons. All right. And the last piece of a nucleotide we need to know is something called a base, which we've heard of before. So there are 4 different options for these bases. And when we call them nitrogenous bases, because as you can see, there's a lot of these letters N, which stands for the element nitrogen. But there are 4 different types of bases that can be on this nucleotide. There can either be an A, a T, a C, or a G. So this one you're looking at right here is adenine, but any of those bases, thymine, guanine, cytosine, can be on this nucleotide as well. But in order to have a nucleotide, you have to have all three of these pieces. We have to have a phosphate group, a pentose sugar, and a nitrogenous base. And these 3 pieces are going to work together to form that double helix of DNA that we've seen before. Okay, so let's look at some more details. The phosphate and the sugar, as we just looked at, the yellow and the blue, form the backbone of the DNA molecule, whereas the bases form the stairs. So what are the 4 bases and how does the base pair rule apply to them? We looked at these yesterday. Remember, those 4 bases are adenine and thymine. They always pair together. So A always pairs with T. And guanine and cytosine. they always pair together. G is always with C. So you're never going to have any sort of crossover. It's always going to be A and T and G and C. All teachers go crazy. Now, I told you guys this word yesterday, but I don't think I required you to write it down, but I want to know, what is this word codon? Well, yesterday when we looked at DNA strands, we saw that those bases usually came in rows of 3. So we might have like a section that was like ATC, GCA, TAG. They're always going to come in rows of 3. And that's because this is kind of a secret code that your cells have to understand. And those series of 3 nucleotides or sequence of 3 nucleotides is known as a codon. And these are going to be super important in the next few units because codons actually code for a specific amino acid, right? So I'm going to actually add that, a sequence of 3 nucleotides that codes for a specific amino acid. And this is going to be super important when we start talking about protein synthesis or the creation of proteins. All right. So again, codons are kind of like a secret code or a secret message. They're always going to come in 3s. All right, moving on. Now let's start talking about this process called DNA replication. All right, so we know that all DNA is made up of a sugar, a phosphate group, and one of 4 nitrogenous bases. New cells are formed when the cell divides into 2 daughter cells. For example, cell division in the lining of your mouth creates the new cells that replace the cells that are rubbed off whenever you chew food. But before a cell can divide, the cell must make a copy of all of the DNA in each chromosome. And this process is called DNA replication. And we talked about this before, right? We saw that when cells divide, they have to duplicate that genetic information so that each resulting cell gets the same amount, right? Which leads us to number 4. Why is DNA replication necessary before each cell division, also known as mitosis? All right, well, I just told you, this happens that each daughter cell receives an identical copy of DNA. We've got to make sure that each cell gets the same amount and the same type of DNA to make those identical cells, those identical daughter cells in mitosis. All right, but how does this happen? So the process of DNA replication is shown in this picture on the right. During this process of DNA replication, the 2 DNA strands of the DNA helix are separated. So they're almost unzipped. You can see right here that this is our original strand. And those strands are actually pulled apart. So they're separated, unzipped or pulled apart. And now each of these blue strands that we see is what we're going to refer to as our template strand. So this is what the cell is about to copy to make a new strand. But how does that happen? Well, the nucleotides in the new strand, or I'm sorry, I skipped a sentence. The old strand is considered a template. The nucleotides in the new strand are added one at a time. And each new nucleotide is matched to a nucleotide in the old strand using the base pairing rules. So remember those base pairing rules. G always matches with C and T always matches with A. So one really important player in this process is actually an enzyme called DNA polymerase. And what this enzyme does is he is basically a master in terms of base pairing. This is his specialty. He is a whiz when it comes to base pairing. And so the second that this strand is unzipped, he knows, okay, it's my time to go. I've got to go match these base pairs. And so he's going to scan along this template strand, right? This is the one he's going to copy. And he's going to see, okay, there's an A, I've got to add a T. Okay, there's a G, I've got to add a C. And he's going to go through and add each of these base pairs according to whatever it needs to match up with. A is always going to match with T, G is always going to match with C. And he does this by running along the entire strand and filling in those bases that need to be added. So then at the very end of this process, we're left with 2 complete identical strands. All right. And we'll be talking more about this over the next few days. But we do need to know that DNA replication results in 2 new DNA molecules that are identical to the original DNA molecule. So each of the new DNA molecules has the same genetic information as that original DNA strand. And this is all because of the action of that enzyme I just told you guys about, DNA polymerase. This is the enzyme that helps to make the new matching DNA strand by adding nucleotides one at a time and joining those nucleotides to the previous nucleotide in that growing DNA strand. So he again is going to run along that original strand and fill in the bases that need to match. So let's review. Number 5, each new nucleotide added by the enzyme DNA polymerase is going to match those corresponding nucleotides in the old strand of DNA according to, what are those rules where the A has to pair up with the T and the C has to pair up with the G? What is that called? Oh, base pair rules. But now I want you to basically act or pretend like you are DNA polymerase. So I've got a strand for you on question number 6. I've given you the template strand. So this is the example that you are going to copy. But I need you to pretend like you're DNA polymerase. I want you to scan across this sequence and add the corresponding nucleotides according to those base pair rules. So remember, A always pairs with T and G always pairs with C. So it is your time to shine. Go ahead and pause this video, complete that strand B to replicate that DNA strand, and then press play when you're ready to review it. Remember your base pair rules. All right, so it's our grand reveal. Now you may notice that this strand is separated in sections of 3. That's not a coincidence. Each of these represents a codon. Remember, again, that's that special coded sequence of 3 nucleotides that will code for an amino acid. So let's get to it. Strand B, drum roll please, should have been T, T, A, G, G, C, G, A, T, G, C, T, A, C, C, G, T, I feel like I'm leading a cheer. TAC, and then CGA. Fantastic job. I'm just going to assume you got all of those right. Now, number 7, describe the function of DNA polymerase. So think about what you just had to do when replicating that DNA sequence, and now describe how DNA polymerase does it. All right, so pause this video, answer question 7, press play when you want to review it. So DNA polymerase is super, super important with DNA replication because it has the important job of basically unzipping DNA, so pulling apart those 2 original strands, and then scanning that sequence and matching the corresponding bases in order to duplicate that strand and create 2 identical copies. So it's going to match those corresponding nucleotides according to those base pairing rules. A is always going to match with T, and G is always going to match with C. And now number 8, describe the backbone of DNA. What is it composed of and what is its function? So think back to the very first thing we talked about today. We looked at the 3 different parts of the nucleotide. 2 of those parts make up the backbone or the side pieces of DNA. What were they? Well, the backbone of DNA is made up of sugar and phosphate molecules. Remember, we highlighted phosphate in yellow and sugar in blue. And those are going to combine to form the vertical side pieces of the ladder. So these form the side pieces of that ladder. They hold it steady. They are the backbone. And then along the middle of it is where we'll have the rungs of those nitrogenous bases. So this is where we would have AT, CG, etc. But DNA wouldn't be possible without that backbone of sugar and phosphate holding it steady and keeping it in place where it needs to be. All right, so to summarize, everything we've talked about with DNA replication, this process is super important because it allows us to make an identical copy of DNA. All right. And that is officially all I've got for you guys today. So Using this information, complete your exit ticket. Have a wonderful day, and I will see you guys soon.